Another way that is used to identify DNA is when we get the DNA from bacteria or from the PCR or from different sources, we get a good amount of DNA. We can cut that DNA into small pieces using specific enzymes, and we can run that DNA in a solid phase. I'm gonna explain to you. This process is called gel electrophoresis and Southern blotting. It's called Southern blotting because we are analyzing DNA, but if we are analyzing RNA, it's called Northern blot. For proteins, it's just protein gel electrophoresis. But we can analyze those three different components, the DNA, the RNA, or the proteins, using this technique. So here in this image, we have a setup of what a gel electrophoresis is. We have a solid phase that is called, of a compound that is called, or a chemical that is called agarose. So this agarose, when we mix it with water and we heat it in the same ways that, for example, we do jelly, it dissolves. But when the temperature goes down and is cold, then it forms like a gelatin surface where we can run some samples. So we use special combs to make the holes in the gel, and when it's jellified, the holes are going to become wells, and we can put the sample of DNA with a specific loading dye into the wells and allow the current. We have a buffer that have ions that allows the current from the negative to a positive electrode. The DNA is negatively charged. It's a negatively charged molecule. If you remember, with the nucleotides, we were talking about phosphate groups, and the phosphate groups are negatively charged functional groups. So the DNA is negatively charged. So we have here the DNA in the negative pole, and when the current starts, it's going to start migrating to a positive pole. And this is going to separate because the agarose, when it's in a solid phase, if it has pores, different pore size, so the small fragments of DNA are going to run in the front, and the bigger size, the bigger fragments of DNA are going to be delayed. It's going to be in the beginning of on the gel. So we're going to have a separation of DNA based on different molecular size. We usually put a ladder, or we put a reference to be able to identify what's the molecular size of the DNA that we're running there. But this is what is called gel electrophoresis. So we separate the DNA or the proteins or the RNA in a gel based on the current, and the different molecular size of the fragments are going to be separated. So at the end, this is just another way of seeing it. So this is the gel we loaded here, and it's going to migrate from negative to positive, and this is how we're going to see the different DNAs that are going to be separated. So these are the beginning of the run, but we let the gel to run for longer time, so you're going to have a very nice separation of the different fragments or DNA that are there. If we want to identify the DNA, what we can do, and actually this is possible to do again with RNA and with proteins, we can transfer all these signals into a membrane. That's the process that is called Southern blot for DNA, Northern blot for RNA, and Western blot for proteins. What we do with that is we have a membrane that we use to specifically transfer this gel. We form a sandwich in which we have different components such as filter paper or sponges, because it requires all the system to be humid to be able to have a transfer of the ions from or the current. And we have the membrane, and on top of the membrane we put the gel, and then we close the sandwich again with filter paper and sponges or absorbent paper, and then we put something heavy on top to be able to make a pressure, and then the whole system starts as well with current, and it's going to transfer the DNA or the RNA or the proteins are going to be transferred from the gel to the membrane, and at the end that's more or less how it's going to see. The membrane, there are different compositions or different characteristics of the membrane. Can be nitrocellulose membrane, can be immobilon membrane, or it can be different size of nitrocellulose membranes. So, but at the end, what you see is almost a picture of what you have in your gel, but it's no longer in the gel. It migrates to a piece of paper, let's say so, but this piece of paper is a nitrocellulose material. And what we can do, because we want to identify a specific DNA, so this is a technique that is allowing us to identify specific DNAs. So for that, what we do is design a probe. This probe means like the primers that we were mentioning before in the PCR is similar to that. It's a small fragment of DNA that we construct based on the specific sequence that we need to identify. So this probe can be 15 to 30 amino acid nucleotides. It has a specific sequence that we need to identify that is going to form a hybrid with the DNA, a specific DNA that is in the membrane. And then this probe can be labeled. For example, some of the probes that we use are radioactive. So when we put the probe with the membrane, the specific DNA is going to interact with the probe, not all of them, because not all of them are the same DNA. We have here a mixture of DNAs. So the specific part of the DNA is going to interact with the probe, and then we're going to be able to develop that image or that signal from the radioactivity in a radiographic or photographic film. So this is a way how we can process the whole DNA, to run it in a gel, to transfer the gel to a membrane, and then to be able to incubate the membrane with a specific probe, and the signal is going to be detected with a photographic film. Or there is another way to detect it as well. It can be fluorescence, but what is important here is this is a very powerful technique that allows us to identify specific DNAs. And it could be done from purified DNA, but it could be done as well with the cell extract or bacterial extract, where what we can do is just break the membranes and get the DNA, and to be able to run it and separate it in different molecular size. So this is, again, I have been saying, another way how to identify a specific DNA sequence, and is by using the tendency of the nucleic acids to hybridize with one another. For instance, if we are looking for a DNA sequence that is GCT, then the probe that we design is going to be manufactured GCA. Why? Because C interacts with G, G interacts with C, and A interact with T, and is going to form a hybrid, and we were going to be able to identify this is, for example, that is the sequence that we're going to manufacture is going to have a radioactive label, or is going to have a fluorescent attachment, and we will be able to follow the signal after the hybridization happened in the membrane. So this is just a close view of what I just told you. The arrangement of the sandwich that we do to transfer the gel, the aluminum, transfer the gel. Okay. So this is just an expanded image of what I just mentioned to you in the previous slide, just for you to have a close view of the process, that is the loading of the DNA, the separation, and then the transfer to a membrane. The system to transfer protein is a little bit different, it's different, but the system to do the northern blotting with RNA is very similar to this that is being shown in this image. And as well, as we mentioned, then we can, after we transfer the DNA to the membrane, it can be identified using the chemical probes specific for the DNA. So this is, as you see, it is a very, very powerful technique. It requires, though, that it has a good amount of DNA here, because if not, then the sensibility of the test is not going to be so good. So the more DNA that you can load here, the better signal you're going to be able to identify later with the identification system. Another technique, which is very wide used in neuroscience, is called in situ hybridization. So the specific or the difference with this technique is that the northern the southern and the western blot, you transfer the DNA to membranes and then you work with the membrane. So you work in another system that is out of the organs or out of the system that we work. In this case, in situ, we work specifically with the tissue. So as you see here, there is a brain that it can be frozen or fresh, but what you can do is different slides of this brain, different sections of the brain that can be fixed in this glass cover slip, and then similar to what is happening in the southern blot, what you can do is treat the sample and incubate it with a probe. This probe, usually for hybridization in situ hybridization, is fluorescent. So what you can do at the end of the incubation, at the end of the process, is to place the slide in a microscope and use with fluorescence you can check and follow the signals. So it is possible that you use a small RNA oligos or to do this hybridization, but as well you can do it with antibodies, and that's the beautiful part of this in situ hybridization, is that you are able to follow proteins as well. Antibodies are molecules that our system or any animal system can generate when a protein or when an exogenous molecule is incorporated in the buffer in the body. So these antibodies generally goes against specific proteins, and we can buy them in different companies, or we can make them ourselves in using mice or rabbits or goats. There are different animals that we do immunizations of proteins or fragments of proteins, and the system is going to generate antibodies against that specific protein. And then we can use that antibody that is purified to incubate with the slide that we have here. Is going to interact with the specific proteins, and we're going to be able to localize in this slide, in the part of the brain, specifically where is that protein present. The beautiful part of the antibodies is that, and as well some chemical probes to RNA or some other synthesized oligos, is that we can put fluorescence, and we can put, for example, the green fluorescence or red fluorescence or yellow, and we can do different antibodies for different proteins at the same time. So those, if you recall or you have seen those images where you can see the cells but different colors, the yellow, the blue, the red. Sometimes they overlap. If the proteins interact, they are localized in the same region, they overlap, the colors overlap. But if they are not close to proximity, then you will gonna see the different spots, different colors in the different regions of the brain. So that's why, because the hybridization happens in the tissue, that's what is called hybridization in situ. And this is a very nice, beautiful technique that allows us to see the distribution and the localization and the regulation of certain proteins or RNAs in the brain directly. It's very important to mention that, as I said before, the antibodies are produced in animals. So these are produced because of the reaction of the immune system in these animals when we make the immunization with the specific proteins or fragments of proteins. So this is a response of the immune system. And because we use this antibody to identify cells with the help or aid of chemical molecules, we call this process immunocytochemistry or ICC. And so we have been talking in these 3 different slides about the southern blot, that is help us to identify different DNA, fragments of DNA. Northern blot, that is basically the same as southern blot, but it's specific for RNA, and the probes that we're gonna use are RNA. That can be radioactive or can any chemical label to be able to identify later in the membrane. And this hybridization in situ that we can use as well to do hybridization with RNA, or we can do immunohistochemistry to be able to identify proteins via fluorescence or via radioactivity as well. So these are different techniques that are, allow us to identify in the laboratory different fragments of DNA. How is the regulation if the DNA goes up and down, or the RNA goes up and down, or protein phosphorylation, or so many different cellular processes where we can use these techniques to identify the regulation and the presence or absence of these proteins in the tissue or in other parts when we use a membrane.